Hello, you are watching single-cell multiome analysis series with SciDAP. In the previous video, we have started Cell Ranger Aggregate pipeline to assemble all our samples in one big dataset, and now we will see the initial result before moving to the filtering steps. The output of this pipeline consists of Cell Ranger summary and the link to UCSC browser. We could click on it, and it will bring us to the 10x Genomics page, where we could see all the statistics about our new sample. Here are the libraries that were aggregated together. You could see there are all our 8 samples. Also the number of cells and median high quality fragments per cell. There's also information about the peaks. It is similar to the report of Cell Ranger count, but now it's all combined for 8 samples. There is also a t-SNE plot that shows you how our cells look like before the filtering. There is also a tab called Per Library, which provides you more metrics about the both part ATAC-Seq and RNA-Seq. For example, the number of reads that were included in the aggregated sample, and the number of cells per each sample. So you could see which samples have more cells and which have very few. Again, the same for gene expression. And this all metrics show you how the sample looked like before filtering, which will be our next step. And here in the files, you could download any file from the analysis that were produced by the pipeline. Also could look at the UCSC browser and explore the UMAP or t-SNE plot. So we could also change the color to the whatever you like. You can change the view. This UMAP looks similar to the one we had as an output of Cell Ranger count for every single sample, but now we see them all together. And it also shows the differential genes. It shows way more cells. Color by dataset to see every sample colored differently, so you could have an idea to what cluster the sample may belong more. For example, this red sample mostly comes to the clusters 5 and scattered here. It is very useful to see for an outliers and for the clusters that might be as a result of the contamination. Also, if you have different conditions like we do, you will see how they cluster differently on the UMAP. And you could download the image, or you could export some cells for the downstream analysis, or just look at the differential genes or the differential peaks if you click on the ATAC dataset instead of RNA. And all of this was the overview of Cell Ranger Aggregate pipeline. And in the next video, I will show you how you could filter your data from low quality cells.